There are over 300,000 species of plants which grow in different habitats or different ecologies, and they have developed certain biochemical and anatomical adaptations to survive these different habitats with low carbon dioxide concentration or high temperature or limited water availability or high irradiance. Plants have considerable variation for the biochemistry of photosynthesis. We have already discussed different pathways for carbon dioxide fixation, that is C3 pathway, C4 pathway and CAM pathway. In this lecture we will discuss some of the biochemical and anatomical adaptations or differences between C3, C4 and CAM plants. In C3 plants, the first product of first reaction of photosynthesis is a 3-carbon compound called as phosphoglycerate or 3-PGA. That is why they are called as C3 plants. Phosphoglycerate. Whereas in C4 plants and CAM plants, the first product of first reaction of photosynthesis is a 4-carbon compound called as oxaloacetate. Because CAM is a variant of C4 pathway or C4 mechanism, therefore C4 and CAM have many similarities. So in C3 plants, the first product of first reaction of photosynthesis is a 3-carbon compound, whereas in C4 and CAM, the first product of first reaction of photosynthesis is a 4-carbon compound called oxaloacetate. In C3 plants, the first carbon dioxide acceptor molecule is a 5-carbon sugar called as ribulose 1,5-bisphosphate. Abbreviated as RUBP. Whereas in C4 and CAM plants, the first carbon dioxide acceptor molecule is PEP. PEP is 3-carbon compound, that is phosphoenol pyruvate. Phosphoenol pyruvate. The initial carboxylating enzyme in C3 plants is rubisco. That is ribulose 1,5-bisphosphate carboxylase or oxygenase enzyme. Abbreviated as rubisco. Whereas in C4 plants, the initial carboxylating enzyme is peptase. The same enzyme in CAM plants. Peptase is phosphoenol pyruvate carboxylase or PEP carboxylase. However, C4 plants have two types of cells, mesophyll cells and bundle sheath cells. The initial carboxylating enzyme is peptase, but in bundle sheath, rubisco is also present. And in CAM plants, we know that They open their stomata during night and keep their stomata closed during daytime. So during night time peptidase is working, whereas during daytime when C3 cycle takes place in cam plants, then Rubisco comes into play. So C3 plants only have Rubisco, whereas C4 and cam plants have two types of enzymes, peptidase and Rubisco. We discussed the phenomena of photorespiration, that is release of carbon dioxide in light in C2 glycolate pathway. When Rubisco acts as oxygenase enzyme, it produces a toxic compound glycolate, and to get rid of this glycolate, plants have to spend energy, and it involves a series of metabolic reactions in 3 different organelles. So in C3 plants, we have photorespiration, whereas in C4 plants this is minimal. Because in C4 plants, Rubisco is only present in bundle sheath cells, and the initial carbon dioxide, the initial carboxylating enzyme is peptidase, which has 1000 times more affinity than Rubisco. And any oxygen that or any oxygenation reaction in Rubisco which results in the evolution of carbon dioxide, that is trapped by peptidase in mesophyll cells. In cam plants, photorespiration can be seen in the afternoon when C3 cycle is taking place. This photorespiration reduces photosynthetic efficiency in 2 ways. Since carbon dioxide and oxygen bind at the same active site of Rubisco, so when oxygen is bound to Rubisco, then photosynthesis or photosynthetic efficiency is reduced. Secondly, there is loss of energy to get rid of the toxic compound, that is glycolate formed in photorespiration. C3 plants, per molecule of carbon dioxide fix, we need 3 ATP and 2 NADH. In C4 we need 5 ATP and 2 NADH, whereas in cam plants we need 6.5 ATP and 2 NADH. In C4 plants we need additional ATP for the formation of PEP or phosphoenol pyruvate, whereas in cam plants we need for the formation of PEP as well as for storage of malic acid in vacuum. However, these plants usually grow in abundant light conditions or high irradiance conditions, so they can form or they can meet the additional requirement for ATP. So C3 plants only have Calvin cycle or C3 cycle operating, whereas in C4 plants Calvin cycle is operating along with HH slag pathway. In both C4 and in cam plants. In C3 plants, the optimum temperature is 15 to 25 degrees Celsius, whereas in C4 plants, the optimum temperature is 30 to 45 degrees Celsius, whereas in cam plants, more than 40 degrees Celsius is the optimum temperature. If we plot quantum yield as a function of temperature, Quantum yield is the moles of carbon dioxide fixed per absorbed numbers or moles of photon. So for C3 plants there is a negative linear relationship. As the temperature increases, the quantum yield decreases, because Rubisco is less active at higher temperature. Whereas for C4 plants, it is a horizontal line. This is the line for C3 plants, and this is for C4 plants. And this is the 25 degrees Celsius temperature. This temperature is 25 degrees Celsius. So what we see is that although quantum yield decreases with increasing temperature, but under lower temperature, in this part of the curve, C3 plants have comparative advantage over C4 plants. That's the reason that still about 85% of terrestrial plants are C3 plants. They have C3 mechanism. But under high temperature conditions, C4 plants have comparative advantage as they have minimal or not detectable photorespiration. If we talk about specialized leaf anatomy, there is no specialized leaf anatomy. They have two types of mesophyll cells, palisade and spongy mesophyll cells. Whereas in C4 plants, there is a specialized anatomy called as Tan's anatomy. They have bundle sheath cells which surround the vascular bundle, and they have their Rubisco in bundle sheath cells. Can plants have succulents. They have thick fleshy leaves or stem with large vacuoles, because in can plants the rate of photosynthesis is proportional to the storage capacity of vacuoles. So they have thick fleshy leaves or stem. But due to limited limitations, they have slower photosynthetic rate or slower growth rate due to limitations of vacuolar storage capacity. So C3 plants only have mesophyll cells. C4 have two types of cells, mesophyll and bundle sheath cells. So they have two types of photosynthetic tissues or two types of photosynthetic cells. Mesophyll cells have package which initially accept carbon dioxide, and Rubisco is present in bundle sheath cells. Can plants only have only one type of mesophyll cells. They don't have the bundle sheath cells. And if we talk about chloroplasts, they have only C3 plants have only one type of chloroplast. C4 have dimorphic chloroplast, and can plants also have one type of chloroplast. Dimorphic chloroplast means they have C4 plants have two types of chloroplasts. In mesophyll cells, the chloroplasts are shorter and they are granule. It means they have granule thylakoids. Whereas bundle sheath cells, they have larger chloroplasts which are centripetally arranged, and they are agranule. They only have photosystem 1. So C4 plants have dimorphic chloroplasts. If we talk about transpiration ratio, then transpiration ratio for C3 is 700 to 1300. In C4 it is 400 to 1600, and in can plants this is 80 to 125. And transpiration ratio is divided by moles of carbon dioxide assimilated. And the reverse of this transpiration ratio is water use efficiency, that is moles of carbon dioxide assimilated divided by moles of water transpired. So transpiration ratio is lowest for chem plants, or they have the highest transpiration efficiency. Then we have carbon dioxide composition point 30 to 70 for C3 plants, 0 to 10 for C4 plants, and 0 to 5 for chem plants. Carbon dioxide composition point is ambient carbon dioxide concentration at which rate of carbon dioxide uptake by photosynthesis is balanced by the rate of carbon dioxide evolved in respiration. So since C4 plants have minimal photorespiration and chem plants keep their stomata closed for most part of the day, so carbon dioxide composition point is lowest for C4 pathway. If we talk about light composition point is the light intensity at which the rate of photosynthesis equals the rate of respiration. So C3 plants have higher light composition point, whereas C4 plants on the other hand have lower light composition point, LCP. It means that in high light conditions, under high light conditions, C4 plants have comparative advantage. They are rarely light saturated. They can better make use of excess light conditions. Some of the examples of C3 plants are wheat, rice, soybean, sunflower, cotton. The examples of C4 plants are sorghum, maize, quinoa, sugarcane, and some weeds like Epiphytes or amaranthus. And the examples of chem plants are pineapple, agave, orchids, and different types of cactus. If we talk about ecological significance of pathway variation, C3 pathway was the first pathway that was biochemically characterized. C3 plants or C3 pathway is the oldest and still the most widespread pathway. About 85% of our plants still have this C3 pathway. Why is that? Because they do not need specialized anatomy, and their energy requirement in terms of ATP per mole of carbon dioxide fixed is lower than C4 and chem plants. And if we see this from altitude perspective, some researchers surveyed the prevalence of different species, and as the altitude increases, the number of C3 species also increases because of lower temperature at higher altitude and higher precipitation. So under low temperature or under conditions of adequate water availability, C3 plants still have advantage over C4 or chem plants. C4 pathway is more suitable for stressful environments because photorespiration is considered the primary selective force for evolution of alternate pathways like C4 and CAM pathway. Since they can concentrate carbon dioxide in their stomata because of Crane's anatomy, and because they can keep their stomachs closed for most part of the day, and due to lower carbon dioxide compensation point, they are better adapted under hot, dry and saline conditions or dry saline conditions. The disadvantage of this pathway is that they have higher energy requirement, but usually they grow under high light conditions or high irradiance conditions, so they can meet the higher energy requirement. So we typically see these C4 plants in tropical and subtropical regions and savannas. So they can. Another advantage of C4 pathway is that they can also grow on relatively low fertility soils. Because for the same water nitrogen content, they can achieve higher rate of photosynthesis, 50% of total soluble proteins in the leaf. Whereas in C4 plants, the concentration of rubisco is 3 to 6 times less than C3 plants. So they are suitable for hot, dry, saline, highlight or low fertility soils. So C4 pathway is an adaptive mechanism to stressful environments. If we talk about CAM pathway, which is a characteristic of arid environments, they have the highest transpiration efficiencies. Their transpiration efficiencies are higher than C3 and C4 plants, but they have low rate of photosynthesis or slower growth. There are two main limitations. One is the limited vacuum storage capacity, because they need to store malate during night time, and second is the high energy requirement, because they need 6.5 ATP under ideal conditions per mole of carbon dioxide fixed. But CAM plants have evolved some interesting mechanisms. There are some obligatory CAM plants like cactus, which only have this CAM pathway, but there are other facultative types of CAM plants which have C3 pathway under optimal conditions of water availability or the absence of salinity, like ice plant, but they can switch to CAM pathway when they come across the stressful environment like low water availability in the root zone or high saline conditions. Another interesting case is that there are some aquatic plants that make use of this CAM pathway. In low carbon dioxide environment, under submerged conditions, they close their stomata during daytime, but they fix carbon dioxide by CAM pathway at night time. This carbon dioxide is released during the process of respiration by other aquatic plants, and these CAM plants can make use of this respired carbon dioxide. And even some CAM plants like cactus can survive for long periods of drought. What they do is that they can retain and assimilate or use the carbon dioxide that is released within the cell during the process of respiration. One more interesting aspect of CAM distribution is found in tropical environments which have high number of CAM species. Uh, they have epiphytes. Epiphytes are plants that do not have their roots in the ground, but they grow on other plants, primarily on trees. So they frequently lack a ready supply of liquid water, but many epiphytic species in tropical and subtropical regions, for example, orchids and bromeliads, can exhibit the camp pathway. So, under cooler temperate conditions, uh, C3 plants have an advantage. Under hot, dry, saline, or uh, high light conditions in tropical, subtropical, or savanna regions, C4 plants can adapt. They can achieve higher level of productivity, even low fertility soils. And under arid conditions or desert environments, then can plants have uh, a comparative advantage over C3 and C4 plants as they can achieve higher transpiration efficiencies. So, in this way, the biochemical and anatomical differences between C3, C4, and CAM plants uh, determine in which environment which type of plants would grow. So still 85% of our plants are C3, about 3% are C4, and more than 7%, that is about 7 to 10% are CAN plants.